months since we have started with the Science Talk Show, and we met many successful, inspiring guests on this show, which pleased us a lot. After this accomplishment, it's my honor to have a person who taught me during my master's. In fact, he's a person many students like me used to consult for solutions. He's none other than Abhishek Mishra, currently pursuing his postdoctoral in Methodist Hospital, Houston, USA. Without any further ado, let's welcome him. Hello, sir. Welcome to the Science Talk Show.
Thank you, Sobin.
So, I know you have done a lot in your life and you have achieved a lot. And in fact, your journey began from India and right now you are in the USA. So would you like to tell something about your journey?
Yeah, it's been a long journey. So research actually started when I started my PhD at National Chemical Laboratory. So um, even before that, when I was a graduate student at Banaras Hindu University, um, I got opportunity to meet some really good scientists at that time. One of them was Dr. Lalji Singh. He used to visit our Department of Zoology in BHU and give talk to us. And that was the period of time when human genome was out and everybody was very excited about it, about the discovery and kind of knowledge it reveals to us. And so that is the point when I realized that I'm going to make a career in research.
Wow.
So I did my master's from the same department as you did in Goa University, Marine Biotechnology department. And then I did my PhD from National Chemical Laboratory, Pune. So there I got an opportunity to, so in the period of this, where I got opportunity to do some summer trainings and MSc dissertation, etc. I formed interest in infectious disease. So I joined Dr. Sarkar's lab in National Chemical Laboratory. This lab was interested in understanding bacterial physiology as well as drug development program. So I, with my some preliminary results and some already existing results from the previous lab members, we realized that there is area which need to be addressed in terms of bacterial physiology, which is the redox regulation or reactive oxygen species regulation to the bacterial physiology. So that time I was working with model organism, which is not infectious for as such for tuberculosis, that is Mycobacterium phlegmatis, and Mycobacterium bovis, which is for bovine animals. So I did with this, and then I moved to Goa University after my PhD as an assistant professor. There I was teaching for 3 years, and I still had this interest of in infectious disease, particularly in innate immunology. So when I was there at Goa University, I wrote a review with my present lab, the current lab where I'm working, on our new understanding about the innate immune system and how it can control tuberculosis. So 3 years later, when I got this position, I was really enthusiastic. Also, I have not worked with biosafety laboratory level 3. So I find this opportunity very interesting. Also some stem cell research, which the lab which I'm working right now. So the basic belief of the lab is that the tuberculosis can be controlled 90% of times with basic innate immune response. So if we can train this innate immune response either with some of the adjuvants or through vaccines, what we can do is we can get a robust protection against tuberculosis. So that's how it's going on, and currently I'm working here.
Yes, sir.
on this.
And in fact, I heard that you got media coverage for one of your research, which you published a few months back, and that is, I think, the novel strategy for tuberculosis vaccine. Can you briefly explain about this research?
Yeah, sure. So we already have a vaccine for tuberculosis, which is BCG, and I'm sure you also have it. I also had as a child, and most of children in India and other Asian countries get this vaccine. Also in Africa, this is a century-old vaccine. The controversy with this vaccine is that it provides a protection which people say is from 0% to 80%.
Okay.
So it's not a very robust vaccine and very reliable vaccine. So there is a need to develop a tuberculosis vaccine. So BCG is basically bovine strain of Mycobacterium. So Mycobacterium tuberculosis is the strain which causes tuberculosis. So the bovine strain is Mycobacterium bovis, and BCG is the particular strain of this species which is used as vaccine. Uh, but the presentation of these antigens is not as good through BCG. So the strategy what we used is we use adenovirus, which is a bovine adenovirus. Like if you have heard about the Covishield vaccine of coronavirus,
Yeah.
for that they're using a chimpanzee adenovirus code, and
Okay.
they code DNA in that vector and deliver it. So we use a similar strategy. We use bovine adenovirus instead of chimpanzee adenovirus, and we coded some of the proteins of Mycobacterium tuberculosis. And what it does is that it induces immune response in the human system. So we did it with some macrophages study with human cells, and basically this whole data is about mice system. And currently we are working on non-human primates like monkeys. We
Okay.
are testing this vaccine, and we got very interesting data from mice. So because we found that the bovine adenovirus coat also produce a substantial response, and which enables my system uh, to to fight against tuberculosis. Now, why it is interesting? So it's not the tuberculosis protein, but the viral coat itself, which means that one of the viral vector, it's diversely affecting other bacteria too. So if it is providing protection against tuberculosis without exposure of tuberculosis, so this is the idea which I told in the beginning, that we can train our innate immune system, which is not very specific, but it can provide a robust response. So these are the some basic ideas which we are working, and we are building up our current research.
So, sir, efficacy is much better than the currently available BCG vaccine?
Yeah, it won't be in absolute sense, because the data which we have is a mice data. So mice and human are not same. We all know that, uh, and humans show some resistance to tuberculosis infection naturally, like in high burden country like our country in India. Uh, we know that there is a heavy burden of tuberculosis. Infectious people are around, but not everybody is getting infected. So one of the reason is that to some
Yes.
extent we are able to have some protection against it. But when we see this in mice, if you infect it with tuberculosis, they don't survive. So their immune system is weaker than the human immune system. So at the mice level, yes, if we compare to with BCG, our vaccine provide a better protection. But the human, we don't know. So that's why we are uh, running an experiment with non-human primates. And if we get a similar kind of data, then we will be expecting a much better response in human, because it is more advanced immune system, and it may provide a better protection. Yeah.
Okay, sir. With that, I remember with the adenovirus and the COVID, since you said you mentioned about the COVID, I know that you have also published one paper related to the COVID, and considering the fact that the COVID have not got eradicated, only the cases have come down. What do you think? What prevention methods and strategy for the treatment you mentioned in your paper, which can be useful for the people? Would you like to share some of them?
Yeah, so the paper you're talking about is 1.5 year old, when we don't have much data about COVID as a disease. Also, there was no vaccine approved at that time. So what we did is we reviewed all the strategies which people are trying at that time, like some of the drugs, which and what are the mechanism of these drugs, and how similar kind of drugs can be developed, and the vaccines which later got approved, like Moderna and
Yeah.
Pfizer. So it was a compilation of all these thing, and that time we have no idea, but now it's a
It's quite.
completely different scenario.
Yeah, yeah.
So one of the thing which is very consistent with the vaccines, that wherever we have 2 day doses of this vaccine, uh, people are able to contain this disease. And also, very important aspect of this is that the hospitalization rate, or the disease severity, which we say, that in which patients are going to the ICU, that scenario is pretty much contained after 2 doses of vaccine. Also, there are two drugs. One is a drug from Pfizer and one from Merck.
Yeah.
They showed a very good potential. They still in the phase of approval, but I'm sure they'll get it very soon. They have already published that data, which 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 shows that there is a sufficient protection. So,
Yeah.
using this in current scenario, I'm hoping that it's getting better, like every month. One of
Okay.
the problems still we have is vaccine discrepancy. Like the countries which are rich, the countries which are developing and in good shape, like even in India. So
Mm-hmm.
the vaccine distribution is very good, but there are many poor countries. They don't have their own resources, and there is a huge vaccine discrepancy. So in the countries which we say developed countries, they are providing booster doses for this vaccine, whereas
Mm-hmm.
some of the countries which have not received a single dose also. So
Yeah.
this discrepancy may be a threat later, uh, because um, this is something which we have already seen, that this virus evolve. If we want to contain it, we need to vaccinate people globally, irrespective of from which country they are, or what are their pursuits.
Yeah.
So that is one of the thing which I think need to be addressed urgently. But otherwise, I think things will get better. After every month, it will get better.
Yeah, definitely it's getting better, and the things will improve soon. So I know you have very good interest, and in fact you are very much enthusiastic about the innate immunity system and on research related to that, and you are currently working on that. But I know one more fact, that you have also worked on nanotechnology. So would you like to tell your thoughts about the nanotechnology and its impact on today's world?
So in so I was I had an exposure of nanotechnology, as I said, when I was doing my PhD in NCL, and NCL was one of the institute which is forefront of nanotechnology. In 90s and in 2000, beginning of 2000, I think there will be hundreds of publication from NCL in that period, in the beginning of nanotech era. I have an exposure. I have been meeting, because these are like neighboring labs. I was interacting with people, so I have some understanding. But it get better when I joined Goa University. They're one of I kept one of my research interest as nanotechnology, and I was also teaching nanobiotechnology as a
Yes,
course
I know.
there. Um, so I designed some of the MSc dissertation topic related to it, and I found that I got very good MSc students. They worked very hard, also very focused, and we were able to mark some progress in that, some good data which was publishable. So that actually increased that interest in nanotechnology. So in my present lab also, now I'm trying to develop one more approach, which is still very nascent, but um, I can talk about it, is that developing a kind of nano vaccines which can stimulate this kind of wide range response, which I was talking about, like a battery reactor. So
Yeah.
as you are a student of nanobiotechnology, you know that even
Yeah.
some of the biomolecules, like for example, viral vector, could be considered as a nano carrier, because of their size of less than 100 nanometer, or little larger than 100 nanometer. So in nanometer range, uh, because these particles are there, so or there could be some lipid particles, or there could be some carbon composites which can be functionalized with some groups, which can induce an innate immune response. So this is one of the site which I'm working on currently also. Also, I
Oh,
saw your video on your channel on nanotechnology. I liked it. It's very
thank you,
good.
sir.
I will recommend your viewers to have a look at it, that on that video
Thank you,
too.
sir.
So, um, so nanotechnology is interesting, and you know that when 20 years back, when people were talking about nanotechnology, particularly in the field of biology, so they have made like in our country, like in India, we have a very good facility for chemistry research. So in chemistry research, they have already marked, like they have made all kind of solar cells. They have made materials which are good for space shuttles and all. So all those kind of materials are being made long back in India, where research infrastructure is not that big, but they have did very good fundamental research in nanotechnology.
Yeah.
But 20 years back, if you ask somebody that what is the contribution of nanotechnology in general biology, so people were very skeptical about it, because there were not much products. There are proof of concept, but they are not actual products. But as we know now, that the two vaccines, like Moderna and Pfizer vaccines, the mRNA is coated in a nano carrier.
Nano care.
So it's something now which is very vast, and it's like day-to-day life. Everybody's getting it. So once you're using it as a part of uh, and also, there are many nanocomposites in different kind of cosmetics is used these days. um, uh, the energy efficient cars, like the one which we discuss as electric cars.
Yeah.
So these most of these cars, their battery, their chips, the automated driving, all these are driven by nanotechnology. So at present, nanotechnology is something which we are using very regularly. Same thing in biology also. So like gene editing system, like CRISPR and all. So the delivery of this system uh, into the cells is taking place through these nano carriers.
Yeah.
So many drug release, controlled drug release. Now these things are reality in present world.
Yeah.
So it has taken a long leap in say 10 years in terms of
Mm-hmm.
actual products. So proof of concept was there 20 years back, but now today we have actual products. Also, sequencing. So there is nanopore sequencing, which um, I don't remember if you could recall that when we were there was a lecture of DNA sequencing. There is something called as nanopore. So I used to say that this is going to be a technology in few years. Uh,
Yeah.
in fact, you passed your MSc in 1 or 2 years. That is actually technology which comes. So there is an Oxford nanopore system available for DNA sequencing, which is using same nanotechnology. So
Yeah.
nanotechnology advancements have been huge in last 10 years. So yes, we are all surrounded by it in any field, from agriculture, medicine to space. Everything is it
In
has a role.
Yeah, in fact, the advancement is such that even a common people, the person who does not know anything about the technology, knows about there is something called nanotechnology. So I think
Yeah.
yes, of course, it have spread a lot, and the advancement is huge. Yes, sir. So thank you so much for sharing your research. Ask you one practical question, like since you have right now you are doing the postdoctorate over there, and you have got it on your own, and so I think you must have faced some challenges which you overcome since you are there. So what suggestion would you like to give to the aspirants who wants to study abroad, from the experience which you got in your journey?
First of all, the one thing. So when I was finishing my masters, so one thing I had is that I want to study abroad for my PhD, or I should do a PhD here in India. So there is a confusion, and so in confusion, if some time goes, then it become very difficult to make a judgment. Like I was preparing for GRE, and at the same time, I got a PhD position in NCL. So I become biased toward that, and I didn't pursue it thoroughly. But
Yeah.
uh, if somebody has a plan, they should start a little early. That is one thing.
Mm-hmm.
Uh, then later, also, it should be very specific that what you actually want. It should not be just that you want to go abroad, but
Yeah.
what is your purpose? So for yourself, not for your SOP statement of purpose, but for yourself, what is your pursuit? Why you want to go abroad? Uh, like if you get a good lab in India and not so good lab in abroad, just going abroad will not make, or it is out the field which is not interest you much. So these things first, the person who want to pursue should do it. Second thing is networking. Networking is something which I think is very very important these days. Um, so once you know which kind of lab you want to go, uh, what kind of work you want to do, then you should start networking. Because as a research career, the most important thing, if you ask anybody, it could be good institute, it could be good research topic, it could be the resources you have for your PhD. But the most important thing would be a right mentor, the person with whom you are going to work. Uh, that that mentor should be very good. That is the fundamental thing of any pursuit of research, um, because that is very critical for any aspect of the career. So at that point, if you start networking with right people, trying to understand, not just rush up into any lab, but you want to understand and go, then that thing is very important. So in present age, so if I see it like 10-15 years back, it was hard to approach because there is only one way. You can email them, um, and you may not get any response, which happens most of the time, because they are also overburdened with lots of emails uh, uh, and request, which is not practical. And some of these emails are not very serious, as they have a certain format that I do this, I want to work with you, and this. So they are not interested in that. It would be better if you network through other platforms, and not for getting a position, but getting into that field and having a better understanding. So this may take a little longer time, like you may have to invest 6 months, 1 year extra, but this will be a very good advantage uh, for starting up a research career, like for PhD or even in postdoc. So um, one more thing, which in postdoc is that people start applying early, like for when they are on verge of their submission of PhD. At that time, they have to they should apply to the positions. But at that time, there is one problem, which is like publications. So some of the publications are not through, some you cannot reveal. So at that point of time, and that makes application a bit weak. But in today's scenario, it's a bit different. Like people publish their work in a preprint server, so it is not peer reviewed, but your publication is in open domain. So these preprint servers like Medrix, Biorix, these are very helpful these days. So once you have your manuscript ready, because publication of a manuscript may take 6 months to 1 year after peer review. There could be attempts in multiple journals. So one of the suggestion I would give to a person who is in third or fourth year of PhD, that they should get these studies published on a preprint server at least. So when you are approaching to another professor or scientist in different country, they can read it and they can understand that what was your approach, what kind of study you do, and they have a better clear perspective. So these things are very helpful, I think, for getting a position, PhD or postdoc.
Thank you, sir. Thank you so much for your sharing your experience. Thank you so much for giving your time, and it's been really lovely talking to you. And after a long we met through this interview, so and now my viewers also know you, and they will know that what kind of research you are doing, and they will obviously learn a lot from you. Thank you so much, sir, for your time.
Thank you so much, sir. Yeah. Thank you.
Thank you.